I had a privilege of presenting the first data from our first-in-human phase 1 trial of the first-in-class mRNA-encoded T-cell engager that targets CLDN6 and CD3 in patients with CLDN6 positive advanced solid tumors. What we showed was that this was well tolerated with promising anti-tumor activity and really provided proof of concept for an mRNA-encoded bispecific antibody produced in vivo. The background to this is that BNT142 encoding mRNA lipid nanoparticles are taken up by the liver and subsequently translated into a bispecific that targets CLDN6 and CD3 called RiboMab-2.1. RiboMab-2.1 essentially is able to simultaneously engage both T cells and also CLDN6 positive tumors. CLDN6 is an oncofetal protein that is not expressed on normal adult tissue, but it's highly overexpressed in cancers of great unmet need, for example, ovarian cancer, germ cell tumors, and also other cancer types such as non-small cell lung cancer and endometrial cancer. During the trial, we escalated from dose level 1 to dose level 7 during dose escalation. And we found that BNT142 was well tolerated with mainly grade 1 to 2 toxicities. We did observe some transient cytokine release syndrome and transient rises in liver function tests, but these were all grade 1 to 2 in general. Beyond that, we also observed sustained and biologically relevant concentrations of the bispecific antibody RiboMab-2.1. Most importantly, we also observed 7 patients who achieved partial responses by RECIST criteria in patients who had CLDN6 positive ovarian cancer, 3 of which were confirmed partial responses. So overall, we've shown and provided proof of concept for this mRNA-encoded approach of an antibody that is a bispecific targeting both CLDN6 and CD3 that's produced in vivo.